After prophase, the cells will then segue into what is referred to as prometaphase. It's this gray area, this gray time between prophase and metaphase. What we see is that the sister chromatids, which are above here on my head here, that were attached to each other during S phase, they attach to the microtubule spindle. They attach to it so that the cells can begin getting ready to separate them. You'll notice that when you look at that, it looks like an X. One sister chromatid is attached to the other at the centromeric region. The mitotic spindle attaches to structures within the centromere that are going to house what is referred to as the kinetochore. The kinetochore is going to actually do the work of separating the two sister chromatids. Metaphase. When you look at metaphase, the chromosomes literally line up side by side. Think about it. When you were a little kid, quickest way to separate the class into halves is to have everybody line up 2 by 2. Everybody on the left go that way, everybody on the right go that way. Same thing here. You're going to line up all the chromosomes and their sister chromatids. You're going to line them up down the center, and then you're going to pull them apart. Now, when you look at pictures, drawings, they make it look all nice and neat, as everything's all lined up at the equator of the cell. But as you notice above my head here, this microscope picture, the DNA, the chromosomes, the chromatin has been stained. And you see, yeah, they're lined up, but it's not all perfectly all nice and neat. But they are lined up. They are attached to the microtubule spindles and are now just waiting to be pulled apart. Then you get to anaphase, the physical pulling apart of the sister chromatids. Now, during S phase, the sister chromatids were attached to each other by cohesin proteins. Cohesin proteins glue the two sister chromatids together so that wherever the original goes, the copy goes along with it. Well, during anaphase, that cohesin proteins break down. And this will allow for the spindle and the kinetochore to pull apart the sisters, as you see by the picture here. It's no longer lined up at the equator. The chromosomes are now moving towards opposite ends of the cell. What we now know happens within the kinetochore are motor proteins. They're literally proteins that walk up microtubules, the spindle. As they walk, they pull along whatever's attached to them. Vesicles, organelles, chromatids. As they walk along, the microtubule that they're attached to gets chewed up, goes away. The spindle, as you see on the far left there, the spindles, which are in the red and green. The green are the spindle fibers, the microtubules attached to kinetochores. They are going to shorten as the chromatids are being pulled apart. The red microtubules are going to keep elongating, pushing the centrosomes further and further apart, aiding in pulling apart the sister chromatids. Telophase. Once the sister chromatids have been pulled far enough apart, there will be a signal sent throughout the cell. The nuclear envelope will begin to return. It will begin to wrap around the chromosomes. As you see above my head here, the chromosomes have been pulled apart. They're now going to be folded back together. They're going to be clumped back together, and they're going to be enshrined, enfolded, I should say, sorry, not enshrined, enfolded by the reappearing nuclear envelope. As the nuclear envelope begins to reform, the spindle apparatus gets broken down. The microtubules, the microfilaments, intermediate filaments, the entire cytoskeleton starts to reform. The cell's organelles also start to reform. All so that cytokinesis can occur. Cytokinesis, the cell splitting. Depending upon whether you're talking animal cells or plant cells, it's going to happen in two different ways. You look up at the top. The animal cell, it's stretched because the microtubules have pulled it apart, stretched. And what's going to happen is there are proteins just underneath the plasma membrane. They're going to start to collapse in together, making a smaller and smaller circle. It's going to start stretching and pulling the plasma membrane almost to the point where it tears. It's going to stretch and stretch and stretch as the circle gets smaller and smaller until, boom, instead of tearing, the membranes just reform as two separate membranes. You've done something like this before when you were a kid playing with a balloon. You take the balloon, you start squeezing it, seeing how far you can squeeze before it pops. Same thing here. You can see, far left there, picture of a cell that is getting ready to go through cytokinesis, or it's in the process of cytokinesis. The plasma membrane's being pulled in and pulled in, pulled in, pulled in, and it's going to either tear or reform. Plants don't have to worry about this. Plants, as you notice, the plant cell just lays down a new wall, because remember, every plant cell is surrounded by a wall. Lays down new wall that's surrounded by membrane. Just start laying down the bricks of the cell wall until next thing you know, boom. What was one is now two.